Thank you. So it's pretty obvious that biologic products have revolutionized our treatment of immune inflammatory disorders. These drugs are not new. Infliximab was the first biologic introduced for Crohn's disease in 1998. So that was obviously over 20 years ago. Needless to say, we didn't necessarily do it right with infliximab from the beginning. I'm not sure we're doing it right with infliximab in 2020. Nevertheless, there have been a number of other reference, and let's start using the term reference products or originators that have developed biologic products since then for both IBD or other immune diseases such as etanercept, rituximab for hematologic and rheumatologic indications, and abatacept for rheumatoid arthritis as well. So next slide. What has happened along these 20 couple of years is that less than 2% of Americans use biologics, but they represent about 40% of all prescription drug spending. And if you just took an average IBD patient on a biologic, the cost was approximately $36,000 per member. Now this is 5 years ago. It will slightly be or substantially greater at the present time as we have new biologic reference products which haven't lowered the price of biologics to date. And so one of the issues is that a reference product, a new drug, costs on the order of about almost $3 billion for a company to develop. And by virtue of the process that we're going to be discussing, the biosimilar process, it's been estimated to have about a $54 billion cost savings. Now, of course, that's over 10 years, so that would average about $5 billion per year. And we've really seen this come into play in Europe, which has advanced beyond us. And I'm going to try to limit my editorial comments along the lines regarding this because obviously Europe has a different economic, healthcare economics than we do. And they've been able on a national basis to lower pricing for a number of reasons, including demanding that patients who are on a reference product or new starts be switched to a biosimilar. So that has lowered the price by nearly 70% in a small country such as Norway. Next slide, please. Now, the way that this has happened, obviously, is globally. Both the European agencies and the FDA over a decade ago began to develop legislation because of this rising cost of healthcare, which is obvious to all of us. And the point of the Biologics Price Competition and Innovations Act, the BPCIA, was to create a shortened licensure pathway for biologic products, again, because biologics take up such a substantial portion of the overall pharmaceutical cost period, to develop biosimilar or interchangeable products. Now, it's obvious to everyone here, but none of the products thus far, and will continue to state this, have been designated as interchangeable, and we'll come to that in a few minutes. Nevertheless, this concept of interchangeability is becoming somewhat superseded by third-party payers in the United States who are selecting products within mechanism of action and even different mechanisms of action. Now, the other important factors that have come into play is that the patents for many of these products are about to end. I think most of you are aware that the patent for infliximab, for Remicade, expired several years ago, which actually opened the door for biosimilars to infliximab biosimilars. Actually, Humira is already off patent, but due to legal procedures and changes in the court systems and individual negotiations, the biosimilars for adalimumab will not be marketed until 2023. In the meantime, we're going to see patents for vedolizumab as well as ustekinumab run out in the next several years. At the same time as the patents have run out, technological innovation has expanded within the industry. And it's quite interesting to look at the different companies that are making different biologic products that they're not actually selling. So for instance, I believe that vedolizumab is not actually produced by Takeda, but it's actually produced by another company, but marketed by Takeda. Don't hold me to that specific comment. But there are a number of companies that have both biosimilar products as well as their own originator or reference products. The other aspect is that with these innovations in the production of biosimilars, that has lowered the cost and lowered the time in order to produce these biologic products, including biosimilars. Next slide. Okay, so it is anticipated that, as I've already said, that there will be up to a $5 billion per year cost saving potential. Now, one of the interesting things that's been brought up in other discussions is the purpose of the act is to lower the cost and to, of course, increase access. I don't have this on a slide, but well, actually, this slide emphasizes that the penetration of biosimilars is expanding. But the other thing is, if we're increasing access to these drugs in other words, allowing more people access to biologic products via biosimilars, the cost differential may actually decline because we're increasing the proportion of patients on biologic products. And we need to eventually factor that in as well. Next slide. So as everyone, all of you trainers know, there are substantial differences between, quote, conventional agents or small molecules and biologics. The first is that small molecules are relatively small compared to biologics. They're made in test tubes. They're generally stable with first order kinetics in how they are eliminated. And the reactions to them, both their mechanism of action and allergies as such, are limited to the product itself. In contrast, biologic products are proteins, which are much larger structures. It is very easy for companies to replicate the protein structure, the backbone of biologics. However, what produces the tertiary and quaternary configuration is the post-translational carboxylation, the sugar moieties that coat these proteins, that give it its activity, both its functional activity as well as its immunologic activity via immunogenicity. So you can change the carbohydrates by changing the temperature of the bath. You can change it by changing the concentration of the different sugars. You can change it by changing the pH, and I'm sure there are some other factors. So it is essentially impossible to make a bioidentical. They are biosimilar because, as you'll see, there may be very minor variations in the structure that do not change its function or its immunogenicity. An important distinction between biologics and small molecules is that small molecules are built in test tubes, but biologics are from living cells, whether it's a microbial cell such as a yeast cell or transformed human cells can produce these via the genetic changes that can be put into the cells. The biologics are actually much more sensitive to storage and handling conditions. Indeed, if you take a vial of a biologic and shake it up extensively, many of those proteins will break down into smaller structures within that. So the handling of these is much more demanding than of most chemical products. And then the reactions can be related to both the product and its potential immunogenicity as well as hosts. And we're realizing now increasingly that there are a number of host factors, such as the recent identification of an HLA type that predisposes individuals to develop immunogenicity, not only to biologics, but probably also to vaccines. We all don't respond the exact same way to a vaccine. So a biologic product is going to be sensitive to both the product and the host's immune system. Next slide. So I actually like this slide. Many people don't, but if you, what it shows is an example of the carboxylation on the left and the carboxylation on the right. And Joy, I think if you build on this, there's a circle that actually shows that the cyclical structure is altered on the right-hand side. So it's very similar. Actually, the molecular weight might be identical, but the sugar structures are organized slightly differently. Next slide. So biosimilars are defined as having equivalent efficacy and safety to the originator. There are slightly different terminologies around the world, such that the FDA says no clinically meaningful differences. The EMA discusses no effect on the safety or efficacy, and the WHO discusses similar in terms of quality, safety, and efficacy. So the terminology is somewhat different, but a great deal of consistency within the intent worldwide. Next slide. And guys, I hope you are keeping notes to yourselves as for our subsequent discussion. So again, reviewing this pretty much replicates one of the prior slides that I showed you and went through in detail. The differences between generics, which apply to small molecules in general, and biosimilars. And again, just to emphasize, it would be extraordinarily challenging and virtually impossible to make a bioequivalent. And some of us may want to speak about the fact that the infliximab, the Remicade, that was developed in 1998 and introduced into the market, is not bioidentical to the Remicade in 2020. And one could say the same thing for any of the biologics. Over these 20 years, they have undergone a number of manufacturing differences or evolutions. Each one has been evaluated by the appropriate agency, resulting in a product that has similar behavior as the original. Likewise, biosimilarly, if you open up a vial of any of the reference products and actually sample what's in that vial, you'll see it's not going to be pure infliximab or pure adalimumab. As I mentioned, when you shake up the vial or if it's maneuvered, there are going to be a number of other smaller products, molecules in there that are actually different from the parent molecule. Next slide. So going back, and again, we keep beating this down, but biologics are highly similar, notwithstanding minor differences in clinically inactive components. And we'll go through the process in a minute. So what clinically meaningful differences implies is no differences in the safety, purity, or potency from the originator product. Another implication of this is that the biosimilars have to be administered in the exact same manner as the originator. So you cannot have a biosimilar infliximab that is an injectable. It has to be administered the exact same way as the originator product. Next slide. So the process, as you all are aware, is different in the development of biosimilars from a new drug. The differences are somewhat of a reversal in the pyramid. When one is developing a biosimilar, the pharmacokinetics and pharmacodynamics are established first to be highly similar to the originator product. Once that is established, clinical efficacy and safety is established in a indication for the reference product. For example, rheumatoid arthritis, psoriasis, psoriatic arthritis, Crohn's disease in adults, Crohn's disease in children, ulcerative colitis in adults or children. Because of the concept of extrapolation, the companies, the biosimilar developers, in order to not have to test the drug in every indication, which is for the reference product, which is the biggest cost in the development, allows extrapolation from one indication to all of the approved indication for the reference product. And the third phase of this is going to be post-marketing pharmacovigilance. And this is the somewhat of an uncertain area at the present time because we haven't been highly engaged in this pharmacovigilance process, although I'm certain that the regulatory agencies are, even though we're not very apprised of that process at the present time. Next slide. So this is the difference in the development. For new drugs, the biggest cost is actually in the orange. In contrast, for biosimilars, there is a much more extensive analytic process to first develop the biosimilar product. And of course, what they'll do is they'll have a whole series of potential molecules. They will identify the one that has analytic properties that are closest to the reference product. After that, they will go through a number of functional assays, such as how well do they inhibit a compound such as TNF? Do they inhibit it in a highly similar manner to the reference product? They will assess animal studies, which are challenging, of course, because many of these are human proteins. But the human studies are only necessary in a single indication of the reference product, after which, if it has highly similar efficacy and safety, those can be extrapolated to all of the other indications. Next slide. So you are all familiar with what pharmacokinetics are, which is how is the drug metabolized in the body, including the absorption and distribution, metabolism, and excretion. And then the dynamics, which is what do the drug actually do to the body? Does it reduce TNF? Does it reduce the signs and symptoms of such and such a disorder? So for biosimilars, there has to be a highly similar PK and PD, but in addition, they have to establish the efficacy in a clinical population that is approved for the originator product. Next slide. Okay, so this is a very complicated slide, but the simplicity of it is that when you look at the effect of infliximab across different indications, of course, the assessment of Crohn's, ulcerative colitis, RA, ankylosing spondylitis, psoriatic arthritis, and psoriasis are different. We don't evaluate Crohn's the same way that we evaluate rheumatoid arthritis. So there will be a variability of outcomes across the different disease entities, but the drug, the biosimilar, has to be similar within one of these entities to be extrapolated to the others. Now, because the rheumatoid arthritis and psoriasis population are much larger than the IBD population, those have been the primary indications that the biosimilars have assessed first before extrapolation to other indications. Next slide.
Actually, Steve, can I ask
sure.
a question? Yeah, I didn't really
Absolutely,
understand.
Betsy.
If you go back to the prior slide, I was hung up on what did the 3, 4, 5, 2, 1, 1 mean? You know, the big numbers across the different indications.
So
Good question. I don't know.
It's
treatment effect.
Tina, you know, or Mark?
I think it's treatment effect size, right?
Yeah. And so they chose the conditions where their original product had the greatest placebo difference.
Delta versus placebo.
Tell the difference between the medication and the biosimilar rather than disease-related variables. It is a hard slide, so we'll have to decide whether or not this is something that's easy for a general audience.
Or that the individuals want to. But I think the main point of this is that you know, the effect of infliximab, the differences, as you can see, are going to be different across indications. Okay. And if you can imagine, with all of these indications, these are trials that led to the approval of Remicade across the indications. And obviously, the development program for Remicade, ultimately, it may be $3 billion to get to market, but subsequently with the expansion to other indications also incurs much greater and additional costs. Thank you, guys. Thanks for your help. I called friends. I called a friend. Next slide, for us. So the other thing that everybody worries about is immunogenicity. And as I've tried to emphasize, immunogenicity has been evaluated in the clinical studies that have led to the approval of the biosimilars. So not only does the efficacy the same, the proportion of patients who develop anti-drug antibodies is going to be similar between the biosimilar and the parent compound or the reference compound. One of the results of that, and this comes up in discussions all the time, is that if you are doing therapeutic drug monitoring for one infliximab product, you can do the same assays with the same interpretation for a biosimilar as you would for the parent compound. So if you're using one of the biosimilars for infliximab, I would say however you're going to use drug levels, you would use it exactly the same. The other important counterpart of this is that if a patient develops anti-drug antibodies to a parent compound, they're going to develop anti-drug antibodies to the biosimilar. So if they have an allergic reaction to Remicade, they're going to have an allergic reaction to Inflectra or Renflexis. It would be very much the same. And I guess another subtle impact of that is that if you give a patient, for instance, infliximab, and then they go on a drug holiday and get started on a biosimilar, you would need to treat the patient the same as if they were restarting infliximab, restarting Remicade. In other words, if you're cautious about a hiatus and worried about enhancing immunogenicity because of that hiatus, you would treat the patient the same way as if you were restarting the originator. Now, I'm, as you all know, I'm very cautious in the way I'm stating that because I think many of us do things somewhat differently with restarts and therapeutic drug monitoring. The bottom line is whatever you would do with the originator, you would do with the biosimilar. Next slide. I don't think that matters so much to my discussion. So I don't know, maybe we should move this slide up earlier because I've talked a lot about extrapolation because this is really a key feature of the agency rules across the world, which is if the biosimilar meets all of the criteria for one of the indications, it can be extrapolated across all indications. So there are some subtle differences in this. So for instance, one of the TNF agents has a specific patent on pediatric use. So if the drug is tested in an adult population, it won't necessarily get the separate pediatric indication. I'm aware of one situation with that at the present time. But for the most part, the extrapolation allows the development of biosimilar companies to avoid the cost of clinical trials in every single indication. But there are a number of rules for extrapolation. Number one, the biosimilar has to meet the totality of evidence, which is based on all those criteria, the PK, PD, the immunogenicity, and all of the developmental processes. It has to be demonstrated in a key indication, which we've just reviewed. And also, it has to, the mechanism and pathophysiology of the conditions has to meet the same. So in other words, although there are differences in how we assess rheumatoid arthritis or psoriasis from Crohn's disease or ulcerative colitis, TNF is an important mechanism across the board with these. On the other hand, another TNF inhibitor, such as etanercept, Enbrel, has not met criteria for extrapolation to other disease states aside from those that it is currently approved for. Maybe we'll want to move this slide up, I think. Next slide. Now, as I mentioned, the final frontier of this is post-marketing pharmacovigilance. And this is also mandated in the Congress law for biosimilars. And even the originators have to meet this as well. And so anytime they change a manufacturing process, that has to be reviewed by the regulatory agencies. The pharmacovigilance is also supposed to monitor for immunogenicity. And in order to do this, what the agencies have done is to add a 4-letter suffix to distinguish the different products. So infliximab, DYYB, I believe is the Inflectra Renflexis product, which is the same, marketed by 2 different companies. The ABDA, I can't remember if it's actually Remicade or
Remsima.
it's Remsima. Okay.
And then Renflexis is separate. That's the Samsung.
Sorry, I'm mixing my biosimilar metaphors. I'm dubious that any of us are going to be really paying attention to this. This is more so for the pharmacists or the clinical, specialty pharmacies. It's not exactly clear how this pharmacovigilance is taking place at the present time. As I said, it's somewhat behind the FDA's wall, but we are assured that this is a process that will be ongoing. Next slide. So I think this is my summary slide, that biosimilars cannot be considered generic biologics. The development and approval is based on a totality of evidence that the biosimilar is highly similar but not identical to the reference product. We don't need to independently establish its safety and efficacy in every indication. The manufacturers use a stepwise approach, which is different than a new drug, which intensifies the analytic portion and reduces the clinical trial aspects of it. The small structural differences are permitted, hence they are not bioidenticals, nor can they be better. I didn't mention this earlier, but if it is certainly potential to make a biosimilar improve over the originator. In that situation, and believe me, the biosimilars are very much trying to avoid this situation, because if the biosimilar actually performs better, statistically better by all of the criteria, then it would be a new drug. And that would not meet biosimilar characteristics. and would have to be reassessed as a new drug and hence in every indication. And that would greatly increase the development cost and essentially be developing a new TNF inhibitor that is not identical to infliximab or adalimumab. The immunogenicity is actually different across indications. Most of us are familiar with the rheumatoid arthritis world, where everybody gets methotrexate as a baseline medicine, which reduces the immunogenicity. On the other hand, in GI, we have actually amplified immunogenicity, particularly, for instance, in patients with severe ulcerative colitis who have a rapid loss of drug in the bloodstream and intense process towards developing anti-drug antibodies when levels drop. So they usually pick a indication such as rheumatoid arthritis, but it's still allowed to be extrapolated after a thorough risk-benefit analysis, and then on to post-marketing risk management. That it says physicians and pharmacists remain alert to. I think that in order to remain alert as a physician, you have to be aware that the patients are getting such and such an infliximab or adalimumab product. And I think that that probably for many of us is getting beyond us as we're seeing a variety of different commercial entities proponing one biosimilar over another, and it is likely to switch from year to year. And this comes back to the concept that the drugs are interchangeable. None of the drugs thus far have been termed interchangeable. In order to do that, in order to get that terminology, a clinical study must go through multiple different switches. And most of the studies that you're going to see that Mark is going to present are either primarily single switch studies or randomization studies between two products. In order to be interchangeable, there have to be multiple switches back and forth. And I'm only aware of a single study in a different indication that has looked at multiple switching, but nevertheless, we haven't had the designation of interchangeability. I think one of the bigger questions to discuss subsequently is that our third-party payers have basically taken this concept on by paying for the least expensive product and not requiring an interchangeable designation. So I'm going to stop there. I don't know if am I on time? I'm not even paying attention. Hopefully we're on time.
You are. You're doing great.
So let's chat.
Oh. Sorry about that.
Hey, Steve, you remember
Hey, Ray.
that Medscape symposium? Remember the slide where it showed all the post-marketing changes in the formulation of different biologics? And I think there's something like 15 for infliximab reference since 1998. That might be a good slide to include, just because I think it's reassuring to know that even the reference products we have go through changes fairly frequently without any loss of efficacy. So I think for people not familiar with biosimilars, that might be a good slide to include.
I have no problem with that.
Another point you can make is that U.S. Remicade and European Remicade are different. They're made in different barns, essentially, and different. So apparently,
I think that's another good point.
In different space, it also comes out different. So
yes. Other comments or questions?
I think along the same lines, the idea, and I like the way you presented it. I mean, we've talked about this before, but Remicade in 1998, and I remember seeing a slide, and I know you presented it, but I remember seeing a slide where there were the changes that occurred each 5-10 years, and I can't remember where I've seen that. It was really good. Because if you take biosimilar today and you look at the changes with innovator Remicade, it's basically their biosimilar Remicade today compared to 1998. So it's again, I don't know that we are going to need to educate or convince our audience that biosimilars are okay. It gets more into the switching and the cost and who's paying for it. But still, I think that was a powerful slide.
I mean, you have, find it if you can and send it to Joy, okay?
Yeah, I'll look for it. Of course I have.
Again, this is kind of a template. This is,
Yeah.
I think, more of a starting point. If you guys find mistakes or modifications in these, you know, we've all been through it, but, you know, we get focused on our own things as you go through multiple slides with this.
And Joy, can I ask you a question? I think now might be a good time, just so that we have sort of a brief organizational format for how these presentations are going to go for everyone. You know, do you how long of a presentation do you anticipate that people are going to be giving, and how much of a percentage focus should be on sections 1, 2, and 3? Because obviously it's going to be dependent on the audience. Some of the audience may need more of the technicalities behind the process, and others may need more of the clinical scenarios, just as we're trying to pick and choose how to construct these presentations for the individual programs.
Yeah, that's exactly right. So the purpose of the presentation here is the one that everyone has everything they need to build what they need, depending on their venue and their presentation, so that they have these options, they have all the material. But yes, as we were slated today for each of the super trainers to present up to an hour apiece, we know that downstream, each of our regional leaders will not have that time for wherever they're presenting. They may have 10 minutes, depending on the venue. And so what you have here, once we're able to do, is you have the core structure and the main ideas, but you can include what's necessary to drive home the point you're trying to make in the presentation you're making, depending on the audience. So you may lose a whole section if the point is clinical considerations or having the conversations, as Dr. Ruggiero just pointed out. Then there may be entire sections that you're not going to need, or you only may need a slide or two from each to make a particular point and then move forward. So you are free to make those changes. Also, for the regional leaders, just know that we're hoping that you can each do several of these over the coming year. And so we know that they may change. So what a presentation you do at a particular meeting in January may be different for a different audience, different topic, than a presentation you may do in April, and that you are free to make those changes as needed for those audiences. So it really is, this is material available to you to be able to put together something rich.
Wednesday night, I gave a 20-minute talk to our system, but the day before, a 1-hour talk to the specialty pharmacies in the system. So, you know, we're obviously all going to be flexible, have different audiences.
Right.
Other comments about the that first portion? That's fantastic. Maybe get out of here by noon.
I apologize. I have a
Berkeley?
quick question. Yes, I'm not sure if I heard you correctly, but did you say that some companies have both the originator drug and the biosimilars, and they
Sure.
produce both?
Absolutely.
What would be their incentive to actually produce both when they already have the originator?
It may. What would be? It's always going to be money.
Mm-hmm.
But you know, it's just their Amgen, I think, makes a number of biosimilars and continues. So does Pfizer now. The companies that are actually supporting this, they all make several of their proprietary and biosimilar products.
I don't know if we discussed this later, but along similar line, can you hypothesize why we haven't been seeing some of the cost savings that were expected, and if that doesn't, follow through? And at least in personal experience, I haven't found that
I
it's
think
easier to get for patients.
Miguel is really tuned into this. So Miguel, why don't you, because he's been at two different institutions with different health systems.
Yeah, so I guess it depends on how you define and who is getting the cost savings with biosimilars. So what I think is appropriately or you've indicated is that the cost savings sadly usually does not go to the patient. So that's the key point to this, is the patient's not seeing a cost difference. So I'll give you the UPMC health plan when I was there, and then the Cleveland Clinic, because they're very different. The UPMC health plan, the actual health plan contracted with biosimilar companies to get a reduced cost. At the end of the day, it was the insurance company, the health plan, that actually saved millions. So there actually was a benefit. That's number one. Number two is a lot of the innovator companies, as you know, do discounting contracts with the individual insurance companies. So, and this is public knowledge what I'm about to say, so I'm not going to get in trouble from UPMC. When biosimilar Remicade came out for UPMC health plan, the contracting people actually did a very good job getting the lowest contract for the biosimilar. Janssen came along and undercut that contract by 3% and then gave discounts to the actual health plan. So the insurance company actually took the Janssen Remicade and they kept actually Remicade on formulary. Moved today to Cleveland Clinic, and we switched to biosimilar completely for Remicade in January 1 of 2019. So pretty much everything we prescribe now is done through our specialty pharmacy based on the contracts that they have. And it is actually saving Cleveland Clinic about $3.8 million a year when you look at the total book of business.
So Miguel, explain how that would operate, because we're seeing the same thing at our institution. They're switching to biosimilars, but explain why if Blue Cross will pay for the reference product, but the patient is getting, the pharmacy is going to administer the biosimilar.
So
if all of you, so all of Cleveland Clinic is biosimilar,
correct.
But you've got multiple different payers.
Correct. So to your point, there are, so I think United in Cleveland actually does not approve biosimilar. So when we infuse it, it has to be outside of a Cleveland Clinic location with Remicade Innovator through Janssen, which is completely bizarre. Otherwise,
but we are seeing this now in different modes. So for instance, we as well, I'm sure all of us, have a combination of patients who are infused in your own hospital or your own center, but we're seeing companies saying, no, we want to have it at home or at a private in a non-hospital setting. So
yeah, it's about 85%, at least what I'm seeing, are biosimilar. About 15% of the commercial insurers that don't contract with Cleveland Clinic mandate Remicade, not biosimilar. But for the most part, it is biosimilar. Some of the patients get what is called white bagging. When they come in, they actually white bag the product. They get their biosimilar in one of the infusion areas. But the cost savings to the system is big. For a patient standpoint, if the patient insists on Remicade and it's not within their contract, not within their plan, they will be required to pay a very large out-of-pocket fee. However, if the patient agrees to the switch to biosimilar, then the cost savings for the patient is low, because I think it's something like a $20 or $50 copay. It's actually very low. So in a way, that's a savings to the patient, but it's a little bit of a forced savings. So I don't know if this was more confusing or helpful, but it's extremely, it's sad that I don't think it goes as much to the patient as it should.
Hey, Miguel, may I ask a question? Is the savings to Cleveland Clinic because of a lower average pricing of the drug, or because you're getting a greater return by using the biosimilar?
No, the lower price.
Because, and is this with 340B pricing for the reference product?
No, actually, this is direct contracting. So it's not even the 340B is completely, would be obviously, as you know, very separate. No, it's not based on that. It's just the way the contracts are set.
Because that's how a lot of the originator products are getting by, is because they're offering the 340B pricing. And so that makes it harder, because we've been using the originator products for the most part, because we are getting discounted pricing. And so adding that to the mix, it doesn't necessarily result in cost savings, but if we didn't have 340B pricing, then the return, I think, is greater because it's ASP plus 6 rather than ASP minus 22, and the cost is also cheaper.
Correct. So when so when I was at UPMC, their McGee Women's Hospital was our infusion center, and that was 340B contracting. And so we've infused everything through there. The health plan actually contracted with innovator Janssen, undercut the biosimilar, and the money actually for UPMC that was made off the infusions was pretty high, just to be quite honest about it. So.
One of the other things I just want to reemphasize in this overall cost savings, and I think it's such a key question of who's getting the savings, is, as I emphasized in Europe, what's happening is they have actually seen an increased access to the biologics. So the price, the cost to the country may actually go up, even though the cost for the product can go down. Because of more people taking the product, which is one of the intents of the law, to increase access to these. So this cost savings has a number of potential variables.
So Maryland's a 340B institution, and they were estimating we were going to save 7 figures going to biosimilar, and it ended up being just a couple $100,000 because of all the different payer restrictions and the transitioning out of hospital-based infusion to home infusion. So I still think that the savings, even with 340B, can be substantial. I mean, they argue back and forth over. Janssen argues with Pfizer over, the actual cost savings, but it was having said that, it was still pretty significant at our institution.
And has industry mentioned at all how the allowance of biosimilars has impacted their strategy in terms of developing originator products versus just creating a biosimilar or something else?
I missed that. I'm sorry.
Sorry. So has approval of biosimilars impacted industry's interest in developing originator drugs?
Well, I think, this is not part of this real discussion, but you see what's happening is that the Janssen's no longer marketing Remicade in favor of its newer product. And likewise, AbbVie, just speaking to the 2 big ones, has 2 new products coming on just before Humira is going biosimilar for marketing. So the companies all have what they call a lifetime management of a product, from launch to stabilization to knowing that they're going to be replaced.
But the reality is, there has to be, I'm just speaking as an opinion, there has to be almost a limit on how many different biosimilars to infliximab and biosimilars to adalimumab there are out there, because to a certain point it's adding, how many of the same biosimilar do you need to maximize your cost savings versus adding unnecessary complexity with this question of interchangeability. So right now, I believe there's multiple different forms of infliximab and adalimumab biosimilars. And so do we really need 4 adalimumab biosimilars?
Yeah. Well, we're in a capitalistic country.
Right.
To determine, if someone wants to come in at 50% of all the other biosimilar costs, why shouldn't they be able to do it if they follow that pathway? I mean, there's no limit to the number of generics for an individual company.
And I guess this is where the regulatory aspect is really going to be key, is that if you have excess and you're cutting down the cost, are you compromising quality or safety down?
Well, the difference, Tina, as you know, in the United States, the regulatory authorities are blind to cost.
Yeah.
This cost issue is a Congress issue, not an FDA issue.
Yeah, but I don't think, to Leanne's question, I don't think that the innovator pharmaceutical company production will change or alter toward biosimilars in any way. I mean, it's just the patent life. If the patent life were ever to shorten, then maybe that would be a different issue. But with the patent life being what it is, and you can see the kind of the amount of money over that first 5 to 10 years, it's going to be hard to imagine that the companies are going to come in with biosimilars also.
Hello everyone, this is Norma. I have a question for Dr. Riguero. In our institution, we do not have biosimilars. Therefore, the majority of the payers are approving the first 3 induction doses, and then they want us to refer out to a tertiary or at home. And they are asking if we can change to a biosimilar. Most of the time, is that a possibility now that could be done?
Yeah, and I think Mark in his talk might get into some of the evidence with switching, but that's very common. We have that as well. Well, we have 2 things, either with, we're not talking about vedolizumab today, but it's not uncommon to have a company approve the first 3 induction, then go home infusions, which personally I'm okay with that. The other is that the induction with a innovator and then the maintenance with a biosimilar, as we're going to talk about probably in the next session, is fine. I mean, personally, I think probably even batch switching is fine, but certainly single switching is okay. The question comes up is that if you are in a batch where there are, to Tina's point, 5 biosimilars that are available, and based on the specialty pharmacy and contracting, they have 3 or 4, then it may be every infusion at home might be a different biosimilar. Then the question is, does that matter? We can have a discussion maybe about that later. But to what you're saying, I think that's okay. And we have the same thing in Cleveland.
And I think that we are going to address that, I think, in the 3rd section when we're talking about clinical. But I think a key is just maintaining the transparency overall, because the patients and the providers need to be aware it can't just be a blind switch. But we see that as well at Cedars, where because of the urgency, they'll approve the induction dosing, and then afterwards they have to be moved out because there's additional costs with infusion at a hospital-based center.
And I think because this is apparent of what's going on to the biosimilar companies that are developing these products, this is part of the reason that they're not looking to get interchangeability and doing those large studies, because it does take a big study to demonstrate multiple switches. But if they see that this is going on de facto anyway, according to third-party payers, they really have no incentive to spend the money to get the interchangeable designation.
Yeah, and Ray just typed in, his dogs are barking, but the idea that there's an ACG study or presented at ACG, but there was also studies in the European meetings where they looked at multiple switches and batch switching, and that was not a problem. Sid, it looked like you were about to say something.
Yes, so sorry. Citrate-free Humira, is that part of the biosimilar? Would the biosimilar be citrate-free? Or is that a way to increase the proprietary, or they just are trying to stay on patent for that?
That's a really good question. It is, depending on the biosimilar, I believe, they have used the citrate-free as a comparator.
Okay.
Yeah, my understanding is they did not patent the citrate-free because that was not part of their patent.
Good question, Sid.
And in terms of post-marketing, like black box labeling, et cetera. So if there's, a new black box warning on the original product, does that automatically get applied to all the biosimilars?
The label is identical. Okay, the drug may be similar, but the labels are identical.
Which also makes it nice because the therapeutic drug monitoring is identical. So we don't have to do anything differently.